We're back with David Reich from Harvard to talk ancient DNA and what it reveals about us. To start us off, how do you describe your work?
I'm a geneticist using ancient and modern DNA to map how people were related in the past and how that connects to people today.
Our last conversation really resonated, and you've got a new study that digs into biology, not just migrations. Set the stage for what you set out to learn.
The field long hoped to track biological change through time, but small sample sizes made that hard. One genome is great for ancestry, but it's just one data point for a trait. Now, with large ancient data sets, we can watch allele frequencies move through time to test where selection really acted.
Why focus on frequency change?
When environments shift, new diets, pathogens, altitude, adaptive variants nudge upward, and with enough data, you can see movements too big for chance. The signal is tiny without big numbers, so scale is everything. The standard view held that recent human evolution was quiet, comparing continents, shows little absolute divergence. Our partitioning shows about 98% of frequency shifts owe to drift and structure, yet the remaining sliver reveals widespread real selection.
But if one population replaces another, isn't that selection too?
Replacement can reflect culture or tech, and it moves the whole genome together, which masks locus-specific adaptation. We look across many small place-time windows with minimal mixing and ask if the same allele edges upward everywhere. Big step era swings, for example, are mostly migration, not adaptation.
On results, you report around 7,200 candidate sites at 50% confidence, so roughly half are likely real, right?
Yes, about 3,600 at that threshold, and many more weaker signals sit below it. The genome looks alive with subtle selection, even if most change is demographic noise. Signals cluster in immune genes and also in metabolism, while behavior-linked sites don't enrich because their effects are spread across many loci.
So that's not saying behavior wasn't selected, just that your hits skew to immune and metabolic biology.
Exactly. Behavioral traits are highly polygenic with tiny effects, so we're underpowered there, but other tests show selection touched them too. The big picture is that selection intensified, especially in the last 5 millennia. Farming, crowding, and close contact with animals created new pressures, and immune and metabolic selection ramps up in that era. It suggests a recent environment genome mismatch being rapidly corrected.
Give us a few concrete examples.
A tuberculosis risk variant surged to roughly 10%, then fell sharply in the last 3 millennia, hinting at a pathogen shift. The B blood group allele rose at the expense of A. Skin lightening accelerated about 4 to 2,000 years ago, and traits like hemochromatosis show reversals with stronger swings in the north than the south.
You once saw little selection over a few centuries in African American genomes, yet